Nathan? Oh, yeah. He was sorting it down.
So, welcome everyone to March monthly meeting. Today we will be discussing a very important topic, malaria matters, insights from a leading expert who is Dr. Niamongo. Dr. Niamongo, in his capacity as a senior researcher and lecturer of biomedical sciences at the Technical University of Kenya, and also a certified diagnostic scientist, public health practitioner, and a research associate at the University of North Carolina, USA. He will be discussing with us today about the impact of malaria as a disease, as a prevention, and as the latest development in the malaria landscape. To give more about his research interest in planetary health in research and education, he actually has been a really efficient principal investigator and co-investigator, as he has conducted research on design, screening, and evaluation of putative vaccine candidates for tropical infectious diseases, ethnographic surveys, and other public health and eco-health related thematic areas. Dr. Niamongo has also published over 26 peer-reviewed scientific articles, over 100 citations on diverse aspects of biomedical research and public health. He has also supervised and mentored master degree students and several undergraduate students. He has extensive experience in managing research groups, staffing, recruitment of study participants, research design and execution, and budgeting. In this regard, Dr. Niamongo had led also funding projects on the development and validation of non-human primates as suitable animal models for studying malaria in pregnancy and preclinical vaccine evaluation. And also the characterization of Trichinella zimbabweensis as an emerging foodborne infection and socioeconomic effects of climate change on jiggers infestations in Kenya. He also serves on various advisory and review committees on higher education curriculum development, the use of animal models for biomedical research, and as peer reviewer of various international journals. As a planetary health enthusiast, Dr. Niamongo is excited to reverse the impacts of human anthropocentric behavior and restore the Earth to original glory through research, education, and community engagement. Welcome, Dr. Niamongo. We are really honored to have you with us. Your contribution as a steering committee member of the MRN since its early establishment really is a big honor for us to learn more from your vast knowledge and to allow this cross knowledge exchange between our established MRN members and the new members who has got a lot of potentials, especially in public health research. So back to you, Dr. Nemongo.
Thank you very much, Dr. Mona, and it's a pleasure to be introduced in such a good light because I've done some work on malaria, and you tasked me to lead this session, and I said I will do, and I've prepared a small presentation exactly to show my journey all the way in malaria. Then out of that, then I'll challenge ourselves exactly what can we do as a team so that we can be able at least to put in a brick into solving or eradicating malaria in this space. So I will ask you to allow me to share my screen. So if you allow me to share the screen, then I'll be able to start the presentation. Okay, thank you. I suppose now we are able to see exactly what we have on the screen is an MRN malaria station, and today's date is there. So basically, as a scientist, I've grown over years to look at the 4 thematic areas that excite me, and on these areas, my interest has been to understand the cellular and molecular mechanisms that are involved in influencing disease outcomes, and why some of the populations are susceptible to malaria while other populations are not. So that has been the major research question that I've worked with, and as well as to understand and identify the putative molecules or targets for vaccine development and design, and which I'll show you some of the work that I've done, as well as I've ventured into preclinical evaluation of malaria, TB, and HIV vaccines, as well as using the adjuvant effect system, so that can be able really to improve the immunogenicity and protective efficacy of malaria vaccines, which I've worked on, and as well as majorly to collaborate with others for technology transfer, for concerted efforts towards better healthcare, to improve the quality of life, as well as to enhance preventive measures for all patients in malaria, and as well as infectious diseases. So far, so good. And this approach, these are research questions I've gone through an approach where I've been looking at the development, validation, and utilization of the African non-human primates as models for translational biomedical research in the aspect of host-pathogen interactions, how the host and the pathogen interact, and what are the disease outcomes, as well as doing preclinical evaluation of the safety and the immunogenicity of DNA vaccines and the adjuvant effects of some of the chemokines that can be translated into effective chemokines for human use, and well as translational medicine, translating what we generate in the lab to the bedside. So this has been some of my major interest. And in the first aspect on host pathogen interactions, I've done some work that has shown that this is some work that we did some time with my colleague, where we showed that placental malaria causes adverse pregnancy outcomes for the mother as well as for the fetus. And therefore, we need a versatile model to study placental malaria. And this presented us with the olive baboon, which is an old world human primate that is available in Africa, that really we treat it as a pest as opposed to an endangered species. So they are in abundance. We get them with the good humane collection and trapping mechanisms, and utilizing this kind of platform, we were able to establish that plus malaria localizes itself inside the placenta because it's an immunologically privileged area. Then now the parasites get to evade the host immunity, and inside the placenta, you'll find that the number of parasites inside the placenta, they are 20 times more than what you get on the peripheral circulation. So this informed us to prepare a policy paper where we said that now when a pregnant woman presents with malaria, and when you determine pregnancy in the clinic or using the peripheral blood smear, where you determine the number of parasites that are in blood, so that shows that in the placenta, there is more than whatever that you get on the peripheral circulation, there is 20 times more in, and this really showed that these are key feature in understanding placental malaria in humans, because there's so many ethical issues in doing placental malaria studies in humans. So utilizing the non-human primate is a very good platform that we can be able to utilize in understand placental malaria, and much work has gone into it, and we have moved a front where we are able to establish how the parasite adheres to the placenta, how the interaction occurs, how does the parasite have the effect onto the fetus as well as, so that is work that we are ongoing and done. So as far as now, we are moving into molecular aspects where we are trying to understand the single cell interaction during the parasite and host interaction during infection. So this was an early publication that we did out of it, and it was actually a key publication utilizing the baboon as a platform for. Then I ventured into understanding co-infections, because most of the parasites in Africa, they overlap. There is geographical overlap, and out of this, we showed that bilharzia and malaria, they overlap quite on a great front, and we were able to utilize the same baboons, and they are very good models, because individually we worked with Plasmodium nosai, which is the 5th human parasite, and shown that it is able to establish a clinical profile that is similar what happens in humans with Plasmodium falciparum. Then we've been able as well to establish that baboons will get chronic schistosomiasis, that which will last over a period of time. And schistosomiasis is bilharzia, which is common in waterborne areas. And this is a disease that people get when because when we have poor sanitation. Then we were able to show that there is enhanced innate immunity when there is a co-infection compared to malaria monoinfection alone. So this shows that when you are co-infected with helminths, it is good because you get to be protected as opposed to getting a single malaria infection. And we showed that now the bilharzia infection will be able to attenuate the severity of malaria in co-infected animals, because we're able to establish that animals that were co-infected had less pathogenesis compared to those that were singly malaria infected, because malaria can kill non-human primates in a span of like 10 to 14 days. They're able to die, but those that were co-infected were able to last all over a long period of time. And this is something that is very interesting, because during COVID time, people were washing hands, and we saw that now this affected our health so much, because we were able to get rid of a lot of infections that will really prime our innate immunity to allow us for protection. And the hygiene hypothesis now, this is now where it comes into play. And we see that parasites are good for us because they enhance our way of survival. And I can relate this with this co-infection, because during COVID, in Africa, we do not have so many vaccines available for us. But the number of cases that we saw, they were dismally low compared to the people that live in the first world. And this really shows that population sustainability and presence of parasites and other comorbidities, or parasitic comorbidities, not lifestyle diseases. Parasitic comorbidities offer a protection compared to other persons. And in this, and I ventured where I wanted to really understand the co-infection with the tissue-dwelling parasites, because a lot of work has been done to understand the geohelminth, the one that we get, those that dwell inside our intestines. And I wanted to really know exactly what happens with tissue-dwelling parasites. This is where I ventured with a group in South Africa, and with Professor Kimbari and Professor Mkaratirwa, where we went and looked at the tissue-dwelling helminths, and we used Trichinella zimbabwensis that were discovered by Professor Mkaratirwa, that we need to understand exactly what are the implications of the tissue-dwelling parasites co-infected with malaria. And this really will help us now to be able to control and manage co-infections in endemic areas, because now people are changing the culinary life, because now tissue dwelling parasites, these are parasites that we get, these are foodborne parasites that we get when we consume food. And these days people are taking meat from crocodiles, meat from wild animals and all that, and you find that they are not well prepared. So they will carry the larvae that will cause infection. So out of this thinking, we ventured into tissue dwelling helminths, where first we ventured into first to understand the metabolic and immunologic inflammatory reactions that occur during trichinella infection. So this is singly to understand the immunological response, and we used mice, and out of this we were able to show that the parasite is able to immunomodulate Th1 and Th17 type responses during chronic infection. And out of this, as well as we will further sought to determine the immune responses that are induced, where we are able now to find that there is distinct differences between Th1, Th2, and T regulatory responses during the intestinal phase, when the parasites are inside the intestines, when they have done tissue migration, and when they have established, they establish different immunological profiles. So out of understanding these immunological profiles, so we went further to do co-infection studies. And co-infection studies, what really came out is that the animals that were co-infected had better immunological profile compared to those that were not. And this is work that has been published somewhere else. Then in preclinical evaluation of DNA vaccines, which have done some work in non-human primates. So this really was to utilize a vaccine delivery system, which is electroporation, because after you've injected the vaccine with the syringe and the needle, you need electrical current to push now this DNA into the cells. So this was the thinking, and this was to optimize the vaccine delivery in vivo. And we saw that electroporation delivery system is an effective method to improve immunogenicity of DNA vaccines, which are encoding Plasmodium falciparum service protein 25. And this was to encounter a problem that we have with DNA vaccines, because they are very poor immunogenicity. So if we inject DNA, it's not able to get into the cells and produce a protein that will become an antigen. And with the electroporation, we were able to show that antibodies against the DNA vaccines were able to be enhanced with electroporation compared to those animals that did not receive electroporation. And this now showed that there was effective transmission blocking efficiency, because the antibodies that we generated, we were able to use them in feeding mosquitoes, and the mosquitoes that were fed were not able to transmit the parasite. So this is something that we did, and these are publications that came in 2013. Then out of that, with that interest in DNA vaccines, I ventured into determining the profile, preclinical evaluation of a DNA vaccine for malaria that was encoding a serine repeat antigen 5, which has been touted to be the best antigen ever to be used as a vaccine. But now it has been done as a protein. So in this case, we prepared a DNA vaccine. And in these DNA vaccines, we couple it with BCG, we coupled it with tetanus epitopes, and we combined with either a chemokine 5 or a chemokine 20, which will allow for chemotaxis of immunoregulatory cells. And out of this, we were able to show that this preparation of the vaccine was very effective and was very safe, because it was able to induce a Th1, a Th2, and T regulatory responses. And we did this in mice, and we did this after mice. We proceeded further to do it in baboons, and we saw very interesting results. But the problem with now vaccines is that after you've generated very good results, now you go back again now to start writing to show that what you've done is good as preliminary results. Now to look for further funds to proceed. So that has been the challenge and utilization, and most people do not want to fund this kind of research because they want immediate profit on investment. So this has not been very fruitful in this front. But what I can say is that out of this, we've been able to show that the African non-human primate is a versatile platform for studying disease outcomes, host pathogen interactions in vivo, because we have shown it that in the few studies that I've shown, and there are several others, using YANMO models, we have been further to contribute information on cellular and molecular mechanisms for infection profiles for malaria. We have been able to understand parasite biology in terms of the way it attaches into human cell, into the primate cells, and how it progresses. We've been able to validate the use of in vivo electroporation as a vaccination strategy in boosting plasmid DNA immunogenicity. And out of this, further, we have shown that incorporating microbial epitopes and chemokines to malaria polypeptides has an adjuvant effect to plasmid vaccines. So the information so far generated is vital for vaccine design and development, formulation of adjuvants, and understanding disease outcomes, and as well as to understand why some of the populations are more susceptible to malaria while others are not. So this work really allow us to move forward in research, in vaccinology and research and design, who's been plagued with so many challenges. That's of co-infections, genetics of population genetics, infection exposures, and further complicated by geographical overlap of HIV, malaria, and TB co-infections. Therefore, in the future, we need to utilize the omics for genome-wide identification of conserved targets for vaccine development, for precision medicine and elimination of malaria in sub-Saharan Africa. We have a vaccine which was launched in October 2021, and it's been widely used in children in malaria endemic areas, but it has raised a lot of concerns. So in these concerns, they have identified so many concerns which are very pertinent. Number one, like there are genetic variations of Plasmodium falciparum protein, that is the circumsporocytes protein, and the impact of interaction on human immunoproteins, that is the HLA, and vaccine efficacy. So people are of various genetic makeup. And with this has impacted the way we respond to the vaccine. And these has had a greater challenge on the vaccine efficacy. So longer peptides of Th2R and Th3R CSF regions are varied in various people. So we need to understand exactly how do we now move to precision medicine, making vaccines for individuals in that localization on their genetic makeup. So that is where we need to go, because this wholesale way of doing things really will not lead us into eliminating malaria from sub-Saharan Africa. Then the cost of introducing and delivering the vaccine within the malaria implementation programs has been a challenge. And this is work that were done by Ranju Baral, and these are shown that really there is a challenge in that way of delivering vaccines. The prevalence, determinants, and reasons for malaria vaccine hesitancy among the caregivers in Nigeria. This was a nationwide cross-sectional survey which showed that the caregivers, they are hesitant to take their children to receive the malaria vaccines. So this is still a public health challenge, as well as there was a systematic and meta-analysis that was done on the prevalence of caregivers' acceptance of malaria vaccine. So now the acceptability in for under 5 in low and income and middle income countries, and people really, yes, the vaccine is there, it has a low efficacy, but do we need to go for it? Or we can survive because we have survived before. So would we need to survive? So for us to, as a group now, to understand and move forward, what we can contribute to this space is that we can now venture into understanding the effect of climate change in the distribution and intensity of malaria transmission in sub-Saharan Africa. This was raised in the chat some time back, and we need now, this is something that we can pick up as a group and really try to look at literature and try to understand exactly how climate change has affected the distribution and intensity of malaria in sub-Saharan Africa, as well as we can look at the anthropocentric activities that are creating major breeding sites for malaria vectors. So when we increase the vectorial capacity, then means that we have more transmission, and this is a challenge to malaria elimination in sub-Saharan Africa. We have a heterogeneous risk of malaria around reservoirs, which are related to the impact of wind direction of malaria transmissions. These are spillovers because we have very big dams that have been built in Africa because of the drought and the effect of climate change. Water has disappeared, so we are building big dams. The Chinese are here, they are building dams everywhere, but now the wind directions and all that, they're allowing the vectors to spill over to other regions that had low malaria transmission. So these are the things that we can look at. And when we look at these things, we need also to understand the strain on healthcare systems due to COVID and the impact of global malaria elimination programs. So these are just areas that we can identify. And there was a very interesting publication that I read that is climate change could shift disease burden from malaria to arboviruses in Africa. So we can continue to focus on malaria, but now we are getting arboviruses in sub-Saharan Africa, and these arboviruses, they're the newer future. So we can compare them. We can compare climate change, arboviruses, and malaria as a cocktail to understand exactly how are they moving, and when they have moved together, what impact or what is the parasite burden or the disease burden that we are getting on the population. So so far so good. These are the few ideas that I had to generate on this area. Then out of that, then we can lead the discussion. I think I can ask that we open the session for discussion, but so far so good. That is me, and this what is in the area so far, and those are the gaps that I've been able to identify. Thank you very much for listening.
Thank you very much, Dr. Nyamongo. We really have benefited from all this overview and thought-provoking ideas that you carefully try to pinpoint so that we can find where are the gaps of knowledge that we can tackle as a team here interested in conducting research on malaria. With this holistic perspective on planetary health and climate change as well. So with this, I would like to open the conversation for a Q&A session. Please raise your hands if you have any comments or any questions to Dr. Nyamongo. So if there aren't any questions now, then perhaps Dr. Nyamongo, you can just like wrap up or summarize the gaps of knowledge that we need to tackle. And yeah, I can see Chanel. Yeah, Chanel, you can go ahead. And then Habiballah.
Thank you so much, Dr. Mona, and also to Dr. Yambongo for that lovely presentation. So obviously, most of you may know that I have a social science background. So I just wanted to have a bit of understanding on when you said that the what is that the species that you were using, that if they had parasites, if they were in fact infected with parasites, then they were not susceptible to malaria. Can you please explain that a little bit more? Like, what does it mean? How were like the parasites being a buffer? How does it work for the species not to be so affected by the malaria parasite? Thank you.
Thank you, Chanel. And what I can say is that immunologically, let's say we have there are those parasites that will influence inflammation. In the body, inflammation is good because it protects you. But parasites, what they do is that they overtly create inflammation. And the inflammation they create is that it affects you now because it has gone overboard beyond the limit. So that's what we call Th1 type 1 immune responses. But also there are these kind of helminths, that is the worms. The worms, what they do is that they influence a type 2 kind of response that is anti-inflammatory. So the cellular parasites, what do they do? They will induce a pro-inflammatory for inflammation. Then these other worms, what they do is that they produce an anti-inflammatory kind of immune reaction. So what happens is that with that counteraction is that, so for the animals that were co-infected, they had anti-inflammatory that was protective against the inflammation, while those that were singly infected they had the pro-inflammatory that went all the way to have overt reactions that really led into even tissue damage. So that is the immunological aspect to that. And when we relate it to the children in sub-Saharan Africa or in endemic areas, if they are co-infected with helminths, then that is good. But after you do deworming, so after you've done the deworming, so you are killing the parasite, the worms. After you deworm, you kill the worms, so the worms will release their antigen. Then it will promote the anti-inflammatory, then they will become protected. So that is the immunological aspect of the protection that we are able to see in that aspect.
Thank you so much,
Thank
Nyamongo.
you.
And Nyamongo, you shed light on a very important topic that because of this mass treatment, for example, the soil transmitted helminths, we are nowadays seeing children with allergy, childhood allergy more than before, because those tiny soil transmitted helminths, or those even giant like the Ascaris, for example, they used to be like protective against the allergy, and this is how the hygiene hypothesis runs around, that in places where those helminthic infections are eradicated, people are suffering from other type of non-communicable infections. So I shall now call on for Habiballah to unmute and ask his question or comment.
Greetings to everyone. Thank you so much, Dr. Mona, and thank you so much for that wonderful and insightful presentation from Dr. Yamongo. I joined the session a bit late. However, I was able to catch quite a lot of the presentation. All right, I just have a question, and I would like Dr. Amongo to make more throw more light on the aspects. I was able to view a slide that talks about the mosquito vaccine. I would like to plead on him to shed more light on the probably, are there any current malaria vaccine candidates in pipeline apart from the mosquito vaccine? Thank you.
Okay, thank you. And what I can say is that currently RGSS is the one that has been licensed for use and is the one that is being used. There are several others that are in the pipeline, and there is one being developed by Moderna using the mRNA technique that they use for COVID. That is what they are trying to champion, but it is still it has not reached where it's been allowed for use, but it's under clinical trials. So far, so good. Mosquirix is the one that is being used, and that's the one that we have in the market. And though there's currently where we're living in this world, we cannot have one vaccine that can be able to really eradicate malaria completely, because malaria is a very intelligent parasite that will evade all the immune responses that are being generated by this kind of portion. Because what they are using is they're using the sporozoite protein to induce, and there are other stages that induce different immunological responses, and then they are able to evade the host immunity. Then they can be able to establish themselves and cause malaria. So that's what we expect into the future, but not at the moment. Yes. But though the vaccines was allowed with very low efficacy rates. Yes.
All right, thank you so much.
Thank you so much, Habiballah, for your question. And it's worth noting that the RTSS vaccine is already in implementation in Nigeria. And this is really bringing a lot of concerns, as it has been given like 5 booster doses, as much as I know, to children. And the structure of this vaccine contains hepatitis B surface antigen among the other micro components. So do you think, Dr. Naimongo, that this raises alarm that the waning immune response also carry a lot of risk, that it's not just like a plasmodium fragment incorporated in that vaccine, but also a viral component? So this is just my question to you, because I think that it really deserves a lot of research before being implemented already in the market.
Thank you, Dr. Mona. And what I can say is that for vaccine design and the new vaccinology these days is that you have to anchor yourself with so many molecules so that they can be able to at least boost a wider spectrum of the immune response. That's why even in my presentation, I showed you that we incorporated BCG and tetanus toxoid in our DNA vaccine, so that we can be able now to make it more immunogenic, to induce a wider spectrum of immunity. That's why they designed the RGSS the same. And this is very good because it gives it a wider spectrum. But now, over time, the kind of immune response that is generated, the parasite will understand it, then will evade the host immunity. That is the challenge that has been there. That's so far why we do not have a vaccine that is efficacious like the way we eliminated polio. That is the major challenge. Yes.
Yes, thank you so much, Dr. Nyamongo, for your elaborations. And let us also hear from Johannes, and then Melvin and Yusuf. Thank you.
Okay, thank you, Dr. Mona. And also thank you very much, Dr. Nyamongo, for your nice presentation. And what my question is going to regarding mental health, since I'm a mental health professional in direct contact with patients with mental illness, because I'm going to develop treatment plan for patients with both mental illness and malaria. And since we know that every or any acute mental health conditions or acute onset of mental illness are caused by any medical condition, even if we cannot rule out their bilateral interaction. So my question is there any consideration we should take or any special attention while treating mental illness regarding malaria for we should take while treating or developing developing treatment plan for patients with mental illness? Any consideration or any attention regarding malaria to develop treatment plan?
Thank you, Johannes, and that is very good because you cast net very wide to include everyone. And from our early evaluation, we were able to establish that there is cerebral involvement during severe malaria infection. So the parasites are able to influence what happens in the brain. So that is a publication that we were done when we were characterizing the baboon as a model for malaria research. We were able to show that there is severe impairment, brain impairment, and even there's some of the work that was done where we were able to replicate the Blantyre scale on impairment or because of malaria. So when you are dealing with such patients, then you need to first understand, do they have infection? So it's just microscopy, rule out malaria infection, then out of that, then you can proceed with the treatment regimen. But if they are both there, so first you have to understand and sort out the malaria first, because malaria causes disorientation, because even the people who have severe infection will be disoriented, because what it does is that it cuts much of the glucose into the brain. Then out of that, then as well as it has so the imozoin that is released will have impact on the brain, and that is some of the work that we did very early, and we were able to establish the cerebral involvement during severe malaria infection. So you have to first treat malaria, then you can treat the other underlying conditions.
Okay, thank you very much.
Thank you very much, Johannes and Dr. Nyamongo. And Melvin, do you have any question or comments?
Yeah, thank you so much, Dr. Mona, and thanks everyone for this amazing session. And I just have a question and also just a comment and some requests from Dr. Nyamongo. We see right now in Kenya, like malaria is shifting also from the lowland areas, like the warmer areas, to highland areas, like where I'm based at in Eldoret, we see like mosquitoes and so on at the moment. And I think there is it has relation to do with the anthropogenic activities happening around, especially during the pandemic, now that a lot of people really cleared land and so on, and maybe the mosquito migrated to these other highland areas. So I don't know if you have some research you can share maybe later on what has been done on that, in that comparison of this malaria in these different regions in relations to climate change. And then another contribution is that we, as a Planetary Health Eastern African Hub right now, through the current project we are working on, strengthening One Health and planetary health in Eastern Africa, we are kind of trying to come up with a planetary health toolbox. And we just have a request. And within this toolbox, we are kind of looking at different planetary health categories, ranging from different topics related to planetary health, in like infectious diseases, mental health, and so on. And we wanted just to request you if you would be interested to be part of the reviewers to look into the infectious diseases happening here within Africa, so that you could contribute in our toolbox. And maybe I could share your details to the other members. That would really be helpful for us. Thank you so much.
Thank you very much, Melvin, and I accepted the invitation, and I'll be very much interested in joining the team because it's for the good of the people. And there is a group that is in Kenya Medical Research Institute that is an entomology group that is in Kisumu, led by Dr. Ochomo, where they look at vectors and the way they are migrating. They're looking at their vectorial capacity. And with that, they've been able to even establish that some of the mosquitoes that are resistant to insecticide are existing in Kenya. So even some of the mosquitoes can travel from various areas, and from what I identified in the areas to look at, I mentioned that due to a lot of development, and we have modified the environment, we tend now to have the vectors increasing their span. So they're able to establish themselves in areas where they were not. So those are the things that really, as a group, we can venture into and try to understand exactly. We can do spatiotemporal analysis. We can do disease mapping and all that. Then with that, we can be able to understand that. Thank you very much.
Thank you very much, Melvin. I extend my thanks to you, and it's really interesting to see that you're joining forces with Dr. Nyamongo to advance with this malaria project, and we really wish you all the best speaking here from the MRN platform that this kind of collaboration has been established. So now I can take the question of Yusuf.
Good evening from Nigeria. I hope you can hear me. Please let me know if you can hear me. Hello.
Hello, yes, we can hear you. Continue.
All right. Thank you so much for your insightful presentation, Dr. Nyamongo. So I really gained a lot from the brief discussion you did. Thank you so much. So I would like to ask a question regarding the effect of climate change on the prevalence of malaria as well as other arboviruses. Just like we've known over the years that the indices of malaria transmissions are mainly two, which are the entomological inoculation rate, which deals with the measure of number of infective bites each person receives per night, and then the vectorial capacity, which actually measures the potential for malaria transmission based on several key parameters of vector populations. So now, having known that the entomological inoculation rate and the vectorial capacity varies with the ambient temperature, which actually initially increasing and then declining with warmer condition. We know that why these two are very important parameters when it comes to conducting a study on how climate change affects the geographical distributions of malaria vectors, and as well as the malaria vectors and parasites. There is a lot of advocacy on the need to adopt a modeling approach in order to understand how climate change is currently affecting the distribution and prevalence of vector-borne disease, such as malaria, and predicting future changes in the risk posed by this disease. So are you aware of any modeling studies, considering the fact that the sub-Saharan Africa actually bears the largest burden of malaria infectious disease? Are you aware of any modeling studies that has been conducted to predict the how future climate change can actually affect the prevalence of malaria transmission in Africa, or if there is no studies on this, what do you think might have affected? What do you think what do you think is actually delaying studies on these key issues? So thank you so much. That's just my question.
Thank you very much, brother, and such a very good, challenging question. And what I can say is that mathematical modeling of malaria transmission, this is something that everyone has endeavored to do. And I'm looking at a mathematical model of malaria. This was something that was done in July 2011. So what I will be of interest is that since you have highlighted the idea, you can share the idea with us, because if you have the expertise and the capability in mathematical modeling, understanding variations, Bayesian and all that, then out of that, you can be able now to understand. But technically, what I can say is that I've not seen a group that is much interested in with the data, mathematical modeling on vectors and all that. So everyone wants really to understand where is the vector, is transmission there or not, then out of that, then they disappear. So such are the things now with this team, with your capability, we can be able to look at.
All right, thank you so much, Dr. Yamongo, for that your answer. Yes, it's something that I've been wanting to look. I've been looking forward to conducting research on it, because I actually discussed this with my supervisors in school, which is actually interested in this area. But it seems like something a bit tasking. But I think as a researcher, we should always be challenged with a task like this. So I'll come up with outline, then I'll share the outline with you. Probably we can come up with a research on this. Thank you so much.
Thank you very much, Yusuf. I'll be very much interested, and I've seen Mona shared a publication on which Yusuf as well, you are leading on it, on arboviruses in Africa, emerging arboviruses in Africa, and this is a very interesting publication, and I can see it's a group on a coffee, so at least this is work that is good done. So if we do such work, then really we will improve our profile and contribute to the community as well. Thank you.
Thank you so much, Dr. Nyamongo and Yusuf. And I shared also this topic of adopting a statistical, mechanistic, integrated surveillance, thermobiology, and holistic approach for arbovirus control in a changing climate. That was our recently published paper also with Yusuf and colleagues. But just Yusuf, you can search for the whole publication on the web and just paste the link there in the chat box, because I didn't have the time to do this. So perhaps now we can have like 5 minutes break so that we can storm on any other questions and then come back with practical ideas to do future collaborative work and research more on this area of malaria. So Vanessa, do you have any piece of music to share with us?
Yes, I will paste in the chat. Does this work? Yeah, does this work if I paste it in the chat and then voila? Okay.
Okay, so it will be just like music because I don't know if Nathan is giving me access to share screen or we shall just play it as music.
Or sure if
Você fez coração, mas sem cuidado, fez chorar de dor. O seu amor, um amor tão delicado. Porque você foi fraco assim, assim, tão desalmado. Meu coração, quem nunca amou, não merece ser amado. Vai, meu coração, ouve a razão, já só sinceridade. Quem semeia vento diz a razão, colhe sempre tempestade. Vai, meu coração, pede perdão, perdão apaixonado. Vai, porque quem não pede perdão não é nunca perdoado. Insensatez que você fez, coração, mas sem cuidado, fez chorar de dor. O seu amor, um amor tão delicado. Porque você foi fraco assim, assim, tão desalmado. Meu coração, quem nunca amou, não merece ser amado. Vai, meu coração, ouve a razão, usa só sinceridade. Quem semeia vento diz a razão, colhe sempre tempestade. Vai, meu coração, pede perdão, perdão apaixonado. Vai, porque quem não pede perdão não é nunca perdoado. Se você disser que eu desafio no amor, sai que isto em mim provoca imensa dor. Só privilegiados têm ouvido igual ao seu.
Okay, so we are back. Thank you so much for sharing that soothing music, dear Vanessa. We always love your choices, and it's really a good one. Thank you so much. And now to some serious talk. I can see that Samuel, Isaac, Percy, Collins, everyone is here. So I know that you didn't join from the very beginning, but then perhaps Dr. Niamongo, you can give like a very rapid wrap up to just put Samuel, Percy, Isaac, and Collins, and everyone coming late to this meeting, just to put their best feet forward and give them a sum up of the research gaps that we need to focus on while trying to conduct malaria research. Thank you. So until Dr. Niamongo comes back, we will need to really make the best out of our time here together. And as you have all shown great interest in different aspects of malaria research, we just need to focus our attention to one single big chunk so that we can try to join forces together and distribute tasks and get engaged in a write-up task and eventually get our collective work published. So I can see Dr. Niamongo joining back again. So Dr. Niamongo, just feel free if you'd like to give a rapid sum up to our members who joined us late so that we can have this solid idea on steps forward.
Hi, thank you. And the wrap up is that to say that we have a vaccine that is recommended, that is in use, but it has concerns. So these concerns needs our input because we are genetically varied. We are from different perspectives. The way we respond to this protein that is being used is different. So we need a vaccine that is more precise for the current generation. That is what we need. And there is cost issue and on implementation that is coming up. That is a gap that we need to look at exactly. And this is something that was done last month to show that really there is a cost aspect. There is a hesitancy, vaccine hesitancy in Nigeria, where caregivers don't want to present their children for RTSS vaccination. There is also acceptance of the vaccine. People are not accepting the vaccine. So what do we need to look at? So those are the challenges. We can peg them. We can put in some information to relook at the vaccine. So those are the challenges. We can, as well as a team, look at the vaccine, try to look at everything that is there. But now that will come with a lot of resistance because now we are trying to bring it down. This is their pet project, and this is the first one. So if we try going that area, you will rub a lot of shoulders. But for us as planetary health people, will be interested in what is the effect of climate change on distribution and intensity of malaria transmission in sub-Saharan Africa. Yusuf has highlighted mathematical modeling and all that. And I, as well as I talked about anthropogenic activities that are creating major breeding sites for malaria vectors. In areas that there wasn't malaria or the vectors weren't there, they are now moving in there. Because in Africa, there are so many dams that are being created. So heterogeneous risk of malaria around reservoirs related to the impact of wind, direction of malaria transmission, spillovers to other regions and all that. Those are areas that are hot now we can look at. Then as well as there's a strain of, the strain on healthcare systems. Because we are in healthcare systems, how did COVID impact malaria, global malaria elimination programs? And as well as to top it up, there is this that was interesting: climate change, malaria, and arboviruses in sub-Saharan Africa, and how it has been combated. Because with the arboviruses, people assume that you have malaria, but in the real sense, you are being infected with arboviruses. So that has been the challenge. So people are learning to treat malaria as opposed to arboviruses, because that's what we are missing. So we need really to understand and look at that aspect. Those are the few gaps that are able to identify out of this. But if you want to start with a new vaccine, well and good. The journey is very long. You will meet so many adults, but where you can see is that now get into the public health space and try now to get information so that you can be able to influence public health policy and practice. Then with that, then you're good. Thank you.
Thank you very much, Dr. Nyamongo. And this really highlighted a lot of areas that we can think together and try to, first of all, put the framework as, do we want to publish our teamwork here as a meta-analysis, a systematic review, or just like something that's really very simple as an opinion article and a perspective article. And again, I'm thinking like we are coming from different geographical areas. We have Vanessa from Brazil. We have Percy from Ghana. Samuel, he's now in Cyprus, and many colleagues from Nigeria, Sierra Leone, Isaac, and so on. We have many MRN members who are based in different locations. So first of all, we will need to know who can take this task seriously, because from our experience, whenever we have a call that we are going to make a write-up with a deadline, and then we try to compile all the fragments so that we can publish them, there's always like some or few members who are lagging behind, and this really affects the overall structure of the whole manuscript after we distribute tasks. So being really serious about taking active contributions is very important. This is the first thing. And then perhaps I can hear also some feedback from some of our members. You know, it's an open talk now, so we can set a deadline, Melvin, but we are not yet with this. So Dr. Nyamongo, can you just suggest like a framework so that we can initiate this work with much enthusiasm and productivity at the same time?
Yes, but now we need to understand it, to identify exactly what area and what are we going to look at before I commit, because sometimes we can look at areas that are not adding up, but it's doable work. Huh? So with that, what are we going to look at? And
Yes.
what are we going to agree on? These are just areas that I identified. They are not absolute. So we can panel with them to fit into the area that we look at. Then from there, we can move on.
Yes, that's really understandable. And from my raw draft that I'm just like speaking spontaneous now, that perhaps we will need to make something like a meta-analysis and then include with this some recommendations that focus on policy making and planetary health approaches, and at the same time these binding global frameworks, how we can commit to it. And I think that with this we can have like a raw blueprint so that we can start drafting something that can be really distributed among interested members who would like to contribute in this malaria research.
That is perfect, and we can agree on what to do. Then we can put it down. Then out of that, we can say this is the time that we need it out, and who will be contributing, and what are the areas that we'll want. Let me write it down exactly.
Yeah, okay. So roughly we can like just vote by raising hands who would like to contribute to this malaria working team on and on researching that area. So let's just try to roughly know who is interested and who is really facing challenges and engaging with so many other stuff and wouldn't really be available for this write-up task.
I think Melvin expressed in the chat.
Yes, okay.
Sure
So thank you, Vanessa.
Melvin, if you are here,
yeah.
please confirm, maybe not.
Yes, I'm interested in contributing, especially aligning the topic of malaria with climate change, because there is a lot of research we've done, but per se under nutrition as well, in the Lake Victoria Basin, and I have some networks. Maybe I will follow up with some articles they've done in that area, and then I would also like to contribute in it.
Thank you so much, Melvin.
Okay, thank you, Melvin.
Okay.
Mona,
yes.
are you interested?
Yes, of course. I'm really interested because from my perspective, the issue of malaria is also gaining traction here in Egypt because there's some cases which is indigenous and some cases which is like from people traveling to sub-Saharan Africa and coming back with those manifestations of fever of unknown origin. It's becoming really frequent. So I would like to have a deeper look at the distribution of malaria because there is a lot of controversy articles around malaria is increasing or decreasing with climate change. So this part, I think that we need to grasp it by like meta-analysis or something like very evidence-based. And you also draw attention on the rise of arbovirus in relation to malaria. But then when it comes to the symptoms and the similarity of them both as febrile illnesses, it's really hard to detect, and it's really crucial when it comes to diagnostic tools. What are the diagnostic tools used so that we can really speak with confidence that this is really relates to arboviruses and not malaria febrile illnesses. So it's kind of, you know, like untouched landscape that I really need to have a deeper knowledge on, and I think many of my colleagues as well. Thank you.
Okay, that is good. At least we have. Then there was this Yusuf. Where is Yusuf? Disappeared. Vanessa.
Yes, I'm here. I'm here. So I'm actually interested. So particularly on the modeling approach to prediction of to identify how climate or weather correlates to the present day distribution of malaria, including other arboviruses. So I'm actually interested.
Great. So I'm not very into malaria, but as Melvin said, of a link with food systems, if we get to this point, I would like to collaborate with this food system relation and climate change and malaria. And I also would love to understand if it's a meta-analysis. I'm not sure if it will be, but since the very beginning, I don't know if you remember, we had a guest with Niamongo that gave us a lecture in MRN, was in the very beginning, about this kind of research. And I'm very interested in that, because I do think we have a lot of information there already, and is missing this, this link with this, the planetary lens. It's very good to produce this kind of work, if you know what I mean. So, yes. Thank you.
I'm sorry, can I quickly make a comment there? So thank you so much, Vanessa. So based on the meta-analysis and then the systematic review that I'll be doing on this research, I think, yes, we're actually progressing. So these are actually some of the research that are actually ranked top in the hierarchy of research evidence. Yes, meta-analysis and systematic review. But Dr. Yamongo, I would like to direct this question to you, because unlike narrative review and other form of research writing, I think meta-analysis and systematic review is actually a little bit complex compared to others, in the sense that we need to conduct some statistical analysis, right? So, and that would involve someone who is actually an expertise in this area, because we cannot decide to conduct a systematic review and start inviting a biostatistician. So that would not speak good of us. So I don't know how we tend to address this issue, because fine, for me, I'm still learning this statistical method. Yes, I'm actually in the process and I'm still learning it. So I'll be learning along the line. So because I won't call myself an expertise in this area. So, and I know you as an expertise, you've written quite a lot of articles or systematic review papers and meta-analysis. So I don't know how we would address this issue, considering the fact that some of us here, we don't have the background of statistics, or maybe we are not really an expert in statistics. So I don't know how we tend to address this issue. Thank you.
May I? Or Wimini was before? It's just about that, if I can make a point. Yeah?
Okay. Okay.
Yeah, it's just, I remember that we also have a statistician, I guess was Mona's colleague in the very beginning, that gave us a lecture or something. Maybe for this, we could invite specifically one person that would, would take care of this part. It's just a thought. Thank you.
Okay, thank you, Yusuf, for your concern, which is very important about the statistical analysis. And I don't really remember, Vanessa, who this person is that I invited, but let us just hear from Niamongo, his feedback on this issue, because it's really one of the cornerstones of conducting a meta-analysis that we need to know how to go around this issue. Thank you, Naim.
Okay, thank you, Mona. Thank you, Yusuf. And Yusuf, you are a good student. And the years I've taught in the university, I always identify a good student, then push them to the end so that they become the best. So now, since you are learning, we will task you to learn with us. And as you are learning with us, we will put you with a lot of pressure so that the output will be seen, because we know what we want. So when you give us exactly what we want, we'll say that now you've matured. So this is very good. And on statistical analysis, so basically a meta-analysis is the overall. Now you want to make a point from everyone has done A, B, C, D. So this is the final outcome from all this. That's that you go out and tell us a story. This is what they did, this is what they did, this is what they did, and I conclude that this is it. But now what we do is that we will extract the data from what has been done, and out of what we are having, I expect to see not much data out of it. So this will be like a scoping review, or actually will be like an analysis that will gather everything, then we'll analyze, then say exactly this is the information that is coming out. This is what is the goal that is, or the information that is cutting across. So to this one, like when you talk about food systems and climate change, you do not expect a lot of numbers. When you talk about climate change and malaria, you do not expect a lot of numbers. So it's not like something that is immunological where you have quantitative data, but this much of it will be like data that we'll be looking at, and we can analyze it systematically, and it's easy to do a qualitative analysis as opposed to a quantitative analysis in a meta-analysis. So Yusuf, you are in here, now you are going to learn, and you shall learn to be the best. Thank you.
Thank you so much, Doctor. I really appreciate that. Yes, I'm ready to learn. Thank you so much.
Thank you so much, Nyamongo and Yusuf. And I can see Yimni's hands is raised. So Yimni, you can just unmute.
Okay, thank you very much, Dr. Mona, for that. And thank you, Dr. Nyamongo, for the great presentation. I actually raised my hand when you asked that whoever is interested in the work should indicate. So I raised my hand because I would be interested. I'm interested in joining this task, and like you all know, I'm coming up. So I really want to learn from you all and put in my very best. Thank you very much.
Thank you very much, Nimni. So now we need to write a list of interested collaborators. And I think that we have like 20 minutes to end this meeting. So is there any other comments or any other things that you need to discuss with Dr. Nyamongo?
Yeah, I just raised my hand to say that I am writing this down.
Yes.
So I'm.
Yeah, thank you so much,
As a
Vanessa.
secretary, yeah.
Thank you so
it's being.
Thank you so much, Vanessa. And I think that we need to be more organized in our schedules so that we can distribute our energy, like personal projects and planetary health webinars and research writing and so on. So it's all about time management, it's all about this willpower. If we really want to write good research, we should have this this kind of being persistent in our, in our ambition towards doing that. So with that, maybe I can hear some perspectives also from Isaac. Isaac is from Sierra Leone, so please introduce yourself, Isaac. It's really a pleasure that you have joined us today, and maybe you can also tell us some of your background, your research background, your professional background, so that we can know how we can collaborate in future projects even. Thank you.
Yeah, okay. Thank you. I hope everyone hear me. Yeah, greetings from Cyprus. I'm from Sierra Leone, but currently I'm in Cyprus, studying at one of the universities. Samuel is my colleague, my good friend here. I know, he's a familiar guy, in the group. Yes, so my background, my first degree actually is in biological sciences, and after that I did a master's in international health, international public health, and currently I'm doing a second master's again in public health, again biostatistics, specializing in biostatistics and epidemiology. Over the years, I've worked in various project health programs, including malaria and other tropical diseases. Well, in terms of research, I participated in a few research projects. I worked for a few years in Burkina Faso with one of the leading francophone research institution that is Centre Miraz, where we did clinical trials, on antimeter, in terms of treating malaria in pregnant women. It was a nice experience. I was there with them for a few months, where again some few field epidemiology experience, and also participating in other researches looking around behavioral change, on different health programs, and on malaria also look at some of the barriers when it comes to the utilization of ITNs, as a strategy to curtail malaria transmission, amongst pregnant women and children under 5. And I've done some few just readings on those kind of stuff around malaria and other stuff. So when Samuel told me about this group, and I think, okay, I think it's another learning point also, and I'm ready and willing to be part of this. But because I'm also engaged here a bit with other academic stuff, so the time sometimes to be part of these discussions was a bit challenging, and this is my first time, to be part of this group, so that's why I've been, like just listening to what was going on. But this is a good, it was a good discussion. It's a good platform. I think it's a right place to be, so thank you.
Thank you very much, Isaac, and this is truly a safe place to be and to engage with all other early career researchers and also mid and late career researchers through this MRN platform. And thank you, Samuel, for inviting Isaac. You're very much welcomed with us, and let us know if you are interested in taking part in this malaria research work, or your capacity now and you have a lot of other obligations, so that we can make a list of potential contributors and start distributing and delegating tasks. Thank you, Isaac.
Yeah, well, really, I'm interested, as long as it's malaria, as long as it's research, it's like I'm always willing and ready to be part of it. But yeah, currently I think my answer somehow filled a lot of stuff. I don't want to put kind of a delay on tasks, because especially when we have tight schedules, and maybe I'm able to meet those deadlines, it puts a lot of pressure on the group, at the end of the day, so for now, I'm not too sure I'll play that active contribution role per se, but yeah, I'll see, where I'm available or able to provide some support, I think, yeah, I'll do that.
Thank you so much, Isaac. And we totally understand, and there's no pressure at all. And it's like a friendly call. And anyway, we are going to also send another email so that members, if they didn't join this meeting, will have a chance to have a say whether they would like to be collaborating with us or we move forward. So we are giving just like 2 days as final decision on who is going to contribute with us. And we can refer back again to Dr. Niamongo so that we will know about the deadline and the general framework of the study we'll be conducting. So now we have like 10 minutes for any closing thoughts. If anyone would like to end this meeting on high note and say anything that can boost our energy or can give us more insights on our collaborative scientific discussions even happened today. So yeah, anyone who would like to say anything can just unmute or we may end the meeting. Thank you.
Oh, excuse me. Let me just make sure of one thing. Could you please confirm? I have here for collaboration for the malaria meta-analysis, Niamongo, Melvin, Mona, Yusuf, myself, and maybe a statistic colleague called Amir Mogzazi. Is that all right? Did I miss anything? I mean, anybody?
Yeah, thank you, Vanessa, for enlisting that. But I don't think that Dr. Amr Mughazi, his name is Amr, because he is originally like a plastic surgeon, and we need someone
Okay.
like really
Okay.
into the field of infectious diseases. But otherwise, I don't think that this should be like a problem now. I think that we need to begin. You know, this
Okay.
is
Yeah,
somehow
I got it.
sometimes resistant, difficult, or we need to gain momentum. You know,
Yes.
so yeah.
Perfect. I will take him out. And about the toolkit, Melvin toolkit, it's Niamongo and Winnie and myself.
Yeah.
Me. Is that right? Anybody else?
Absolutely.
Okay. So thank you. I mean, it's open for any thoughts or any closing thoughts or whatever you feel like saying and sharing. Thank you.
Thank you very much, Vanessa. And we'd like to give note that our April meeting, we will be delighted to have Melvin as the moderator for the session. And she's got a lot of experience in food science, climate change, and many other aspects in planetary health. So we really look forward to our next meeting in April, and perhaps also it's according to Melvin whether she would like to invite collaborators to a potential new research topic or not. But it's just up to you, Melvin, to draft the agenda for the day and get the best out of our gathering together. Thank you so much.
Thank you so much. I will do so. And thanks for involving me in that.
Thank you. It's our absolute pleasure and a true honor that all our MRN members are really actively participating and enhancing our meetings to something like productive, something that can really be beneficial for all of us. With this, I will end the meeting. Thank you so much once again, Dr. Niamongo, for sharing your knowledge, your experiences, and for taking of your valuable time to be with us here and to discuss. We truly we are very grateful to all our MRN members, our MRN Secretariat, Vanessa, and our research manager, Yusuf. and of course our ACOFI founder Nathan. Thank you so much. Thank you, Collins, Percy, Chanel. Thank you so much for joining us. And next month, inshallah, we will meet with Melvin, and we have a lot to discuss on that meeting, inshallah. So take great care of you, and until next time, bye-bye.
Bye.
Bye-bye.
Bye-bye.
Bye.
Wonderful presentation, Dr. Nyamongo.
Bye.